in the future, whole exome sequencing will be indispensable. So you have to do a whole exome sequencing to get more information about the patients. Currently, we are looking on a subset of genes. In the future, we will have a larger comprehensive view on the genome or the exome of the patients. So you get more information. Maybe they are now not treatable, but if you have a look again into the same patient 1 or 2 years later, there might be new biomarkers on the coming, and this means those patients can be directly treated because we already know the information that the patient has something which might be addressable in the future. So, and this is something which is more interesting. So you get more complexity. So you see the pathways which have a mutation, which can be maybe also addressed because it's a pathway which can be addressed and not only the mutation itself. And the other point is that you have a deeper insight into the genomic architecture of the patient. And this is also something which have an influence on the future perspective. So regarding immune checkpoint blockade, maybe there are a high tumor mutation burden which can be addressed, microsatellite instability, but also again you find incidental findings which you are not aware. Maybe you can also find out why a patient does not respond to therapy. And this is something which is now lacking with the targeted smaller panels, so you have just a small perspective of the patient itself. But this is something which will be increasing in the future. For example, in Germany, we have now this model project where we have to, or we will perform whole exome sequencing for all our oncological patients. And this will help the future research on these patients and to find potentially new biomarkers. For example, you can also try to find out or to read out the ADCs, which are already now an emerging new biomarker field. So maybe you can find amplifications of those genes. We can collect more information to get further information about these patients, about their setup of mutation, the mutational profile from those, and maybe also get hints which is the correct therapy for those patients. Maybe they have one mutation in a known gene, but the second mutation which shows that there is no benefit for the therapy. And then you can give the patient the option to get the right therapy upfront and not first the wrong therapy, which might not have a benefit for the patient, and then later when the disease has already progressed, then another therapy. But the most important thing is that we get more information about the profile of these patients. So we know more than about the landscape, about different tumor entities. Also, maybe the histological definition of a tumor entity will change regarding to the molecular profile. We already saw that in the last years for lung cancer, where we started with small molecular profile. Then now with the increasing molecular profile, you see, oh, there are sub-entities. And this might also have a benefit for the future progression of therapy development.